Okay, I think we're good. All right, I'm really excited to be hosting this PolarConnect event with you guys tonight on June 26, 2019. This is Janet Warburton. I'm the project manager for PolarTREC, and my colleague Judy Fonstock is also on the phone. I'm coming from Anchorage, Alaska, and she's in New Hampshire. And tonight we have our PolarTREC teacher, David Walker, who was along with his several members of a research team, but the primary one that's going to be talking tonight is Rose Cory. And they're up at Toolik Field Station, which is, they'll have a map, I'm sure, to give us a better sense of where they are, but it's in Alaska, Arctic Alaska. So with that, before I turn this over to them, there's a few things about the platform and about the program that I want to share with you. First of all, many of you are probably already starting to do this. I can kind of see the chat area has lots of names and people saying hello and where they're from. Thank you for doing that. Keep doing that, and that just gives us a sense of where you're all coming and joining us from. So tonight's program is, and somebody's even doodling. So no doodling, please. This is what happens when you have a rowdy group of family joining. They just get crazy with the things.
100% for doodling.
Yeah, no doodling, okay? Unless it's a smiley face. All right. Dave, can you go to the next slide for me, please? So tonight, well, we're presenting. If you do have questions, and this event is being recorded, so we'd like to capture those questions in the chat log so that others that haven't been part of the presentation can review that and hear the answers. So please type your questions in the chat log, and we'll try to respond to them as we go along. And if we don't get to your question during the presentation, we certainly will at the end, and then you'll also have a chance to ask your questions live. And the reason for this whole program, or the reason why Dave is in the Arctic, is he's part of PolarTREC. It's a program where we put teachers and informal science educators with researchers like Rose in both the Arctic and the Antarctic. And we are funded by the National Science Foundation. And the whole program is administered by a nonprofit that's based in Fairbanks, Alaska, called the Arctic Research Consortium of the United States. It's a mouthful, or ARCUS for short. And our whole goal is to give opportunity like this to educators to work with scientists in the field, learn about what is happening in polar science, and bring that back into their classrooms or to other learning environments. We've been doing this since 2004, and to date we have over 190 educators that have participated in our program. So we're really excited that Dave is one of them as well. And I see some other alumni that have joined us tonight as well. Let's see. Like I said earlier, this event is being archived. After the event, we will share the link and post it on the website and send it out to everybody that's registered. There's lots of PolarTREC things happening. If you haven't been to our website or explored it beyond the PolarConnect web form, there you can follow other expeditions. We have a woman in Siberia right now, and we have other expeditions happening throughout the summer as well as into the winter down in Antarctica. There's an education listserv, and there's some great resources that you can access from our website. I think that's my spiel, so at this point I'm going to turn it over to Dave and Rose. Welcome.
Hello. Thanks everybody for joining in. I know quite a few of you. I don't know everyone, but it's great that we have so many people that are here today to listen to an explanation of Dr. Cory's amazing research up here. The title of this presentation is Carbon in the Arctic, which is really what we're focused on. And I'm going to be specifically focusing on stuff that I've been helping out with up here throughout the month of June. And up here is Toolik Field Station. So we're up here in the Alaskan Arctic, pretty close to the Arctic Ocean. and it's been a really, really fun experience. So briefly, to start off, some more formal introductions since I don't know everybody. Again, David Walker. I'm a high school teacher from Austin, Texas. I've been teaching at Lhasa High School, which is a public magnet school there, for 9 years. And I teach 2 courses. One is entitled Planet Earth, which is a geobiology course that focuses on the history of the Earth from a physical and biological perspective, and the interplay between the physical and biological planet throughout geologic time. There's a significant field work component to that course. Students get out and do projects around Austin at parks and preserves. The second course I teach is for juniors and seniors, and it's an elective in organic chemistry. So I'm really blessed that I got paired with Dr. Cory's team, because their work really threads the line between the 2 disciplines that I teach.
Great, and hi, I'm Rose Cory. I'm a professor in Earth and Environmental Sciences at the University of Michigan. And I've been working in the Arctic for a while here at Toolik Field Station in the Alaskan Arctic regularly since 2010, and also I started, my first year up here was 2002, so for a while. And I work on, or I study the cycling of carbon here in the soils and in the many streams and lakes on the landscape. And as you'll hear more about, the reason that we do that is to understand how a warming Arctic is going to feed back and influence everyone on Earth. And because what's happening here in the Arctic is so important, we're really excited to have the PolarTREC teacher, Dave Walker, up here with us so he can communicate you know, how the biogeochemistry and chemistry that we're doing up here is going to influence, you know, people living in Austin, Texas.
So a little bit more about where we are. Toolik Field Station is about 9.5 hours north of Fairbanks, Alaska, by car. And I say by car, we're in the Arctic. How do you get here by car? They're mostly trucks that drive on this highway. It's called the Dalton Highway, and it's a mostly dirt highway that connects Fairbanks with the Arctic Ocean at Prudhoe Bay. And this highway was built around the time that the Trans-Alaska Pipeline was constructed in the early 1970s. And the Trans-Alaska Pipeline runs from Prudhoe Bay, or the Arctic Ocean, all the way down to southern Alaska. And so we drove on the Dalton Highway. We got in on the 3rd of June at Fairbanks. And then on the 4th, we loaded up in trucks and drove up here 9.5 hours. It was an interesting road, pretty bumpy. A first experience for me, but Dr. Cory's been on it quite a few times. And on the right there, center is a picture of the Dalton Highway. Left is a sort of the route that we took. And then there's me on my first day at Toolik Field Station, and you can see that it doesn't really look like what most people would deem the Arctic. It's very green, skies are often blue, oftentimes you can wear short sleeves outside, so it's been a pretty interesting fluctuating experience up here. Toolik has been around since probably around I think it was the mid-1970s that they started their first research station up here. Pretty soon after they actually built the Dalton Highway. So it's been a center for Arctic research for a very long time. And it's a really cool melting point of science, or not melting point, melting pot for science. Because, you know, at your lunches and dinners, you just interact with a wide variety of people doing all sorts of different types of research from all over the globe. So it's been a really educational opportunity for me coming from Texas. So I want to start off with a little bit of background information that was helpful for me to understand Dr. Cory's research. And the first, probably most important piece to this puzzle is permafrost. And coming from Texas, I had very little knowledge of what permafrost is, so I had to learn as well. But permafrost is formally defined as ground, so rocks, soil, silt, that's been frozen for 2 or more years. So completely frozen, hard frozen for at least 2 years. And I was really surprised to learn how important this type of landscape is in the Northern Hemisphere. It accounts, as you can see on the map there at the right, for 24% of the landscape in the Northern Hemisphere. So it's a huge part of the land in the Northern Hemisphere is this stuff that's permanently frozen. Above it is what's called an active layer, and the thickness of the permafrost layer varies based on location, as does the thickness of the active layer. It also varies based on season, but the active layer, as you can see in that diagram on the left, is a layer that periodically freezes and thaws with the seasons. One of the more important parts regarding permafrost as related to Dr. Cory's research is that it houses an incredible amount of organic material. So this is organic material that is from plants and animals and microbes of the past, and it's frozen and locked away in this permafrost that's been frozen sometimes for 9,000, 10,000 years. So there's a huge amount of organics. How much? Well, I looked it up, and permafrost globally houses 1,600 billion tons of carbon. And that's just a number to you until you realize that's double the amount of carbon that's in our entire atmosphere. So this is a huge carbon source that is just sitting up here frozen in the Arctic. So that brings me to dissolved organic carbon. So what is dissolved organic carbon? Well, permafrost melts, and because of global warming, it's been melting at an unprecedented rate. So what happens? Well, water leaches out the organic material that's in all that permafrost, and it becomes dissolved. And so we're going to be using this term dissolved organic carbon a bunch throughout this talk, and that we're going to abbreviate it DOC so we don't have to say it too much. But DOC, if you ever hear DOC, all that is dissolved organic material that is in Arctic watersheds. Where does the organic material come from? It comes from thawing permafrost. When permafrost is frozen, it can't, those carbon molecules can't become part of the dissolved organic content. The coolest thing that I learned about this dissolved organic carbon is that it has a color. Teaching organic chemistry, most organic molecules appear white. Why do they appear white? Well, they're absorbing light in the UV range, which means that when visible light hits them, it's all being reflected back to the viewer. So they appear white, kind of like a white powder. And most of the stuff we deal with in my lab back at Lost High School, it looks like a white powder. But organic molecules that have a high degree of electron delocalization, the electrons can spread out throughout the molecule, are very unique in that they can also absorb, in addition to UV light, visible light. And when a compound absorbs part of the visible spectrum, it's not reflecting back all of that spectrum to the viewer, which means you see a color. And these materials, just like lignin here on the left, which is one of the primary components of dissolved organic carbon, but it's an incredibly complex mixture of a lot of different things, they have a high degree of electron delocalization. So they're absorbing in the UV and in the visible. So Arctic streams are colorful. And Dr. Cory had this really cool way of thinking about it. She explains it like tea. Just as you can brew different types of tea and those resulting teas have different colors, from red zinger, which looks red, green tea, obviously black tea, chamomile. Different Arctic streams have different colors because of their different characteristic dissolved organic carbon. So very, very cool. This is you.
Right. And so, and I should point out that there's dissolved organic carbon in all waters on Earth. So you all probably notice these in the streams, rivers, lakes, and coastal waters, even around your homes. The difference, or what distinguishes the Arctic, is that we have the streams up here have a lot of dissolved organic carbon. So the more dissolved organic carbon, or DOC, in water, the more colored the water. And why we care about this so much up here in the Arctic is, as Dave said, the permafrost soils contain more than double the amount of carbon in organic form in soils than is in the atmosphere in inorganic form as carbon dioxide. And this is important because carbon dioxide, of course, is a greenhouse gas and acts like a blanket to keep Earth's surface warm. And when we have carbon in organic form, this is carbon in soils. It's that brown color in the soils and the brown color that Dave showed in the water and in dissolved form. This carbon has the potential to be oxidized and turned into inorganic carbon, or carbon dioxide. So this is what I work on, is the cycling of carbon between its organic form, that colored form, and its inorganic form, which is carbon dioxide, and it's a greenhouse gas. And what we're looking at here is what happens when this dissolved organic carbon, this colored material in water, is flushed from soils. And then in soils, as shown, or once it's flushed into surface waters, as you see on this slide here, we have an arrow of DOC coming out of soils and turning waters this golden to brown kind of root beer color. There's two things that can happen to the dissolved organic carbon. One is it can get completely oxidized all the way to carbon dioxide, where it then is released to the atmosphere and contributes to warming, because carbon dioxide, again, is a greenhouse gas. And if all the carbon in these permafrost soils were to be oxidized to carbon dioxide, we would more than double the amount of carbon dioxide in the atmosphere. And if you think about that, that's a huge consequence, because we've gone, you know, from the past 150 years, we haven't doubled the amount of carbon dioxide in the atmosphere. We've gone from 280 to about 400 parts per billion. So this is why we're so concerned about carbon in permafrost soils, is the potential for all this carbon to be turned into carbon dioxide. But it may not be. The other fate or the other pathway is this dissolved organic carbon can be washed out to the Arctic Ocean, where if it's not oxidized to carbon dioxide, it could basically reside in organic form and end up basically in another freezer, going from permafrost soils, one freezer, to the Arctic Ocean as another freezer.
So this is what we call positive feedback. And this permafrost thaw DOC cycle becomes a positive feedback loop. Why is it a positive feedback loop? Well, permafrost is thawing because of global warming at an unprecedented rate. What that does is allows for much more higher concentrations of dissolved organic carbon to leach out into Arctic watersheds. Those organic carbons are broken down by sunlight and microbes into carbon dioxide. That carbon dioxide feeds back in and makes global warming worse. And so this is positive feedback. It's a self-reinforcing cycle. And it's one of the main reasons why global warming is amplified in Arctic regions. If you look at this really remarkable graph or chart of the world prepared by NASA, you can see that, yes, okay, so the baseline, the 0 point, is a baseline average temperature from between 1951 and 1980. And so you can see most of the Earth has warmed since that average. But look at where the highest intensity of warming is. It's up at the North Pole. It's up near in Arctic regions, including where we're sitting right now. And this permafrost positive feedback loop is one of the main reasons why. If you look at the data over the past 30 years, the average temperature in Arctic regions has increased by 0.6 degrees Celsius, which is around 1 degree Fahrenheit. And when you hear that number, you're like, that's not going to affect me at all. I mean, maybe I'll get a little bit more sunburn, but I mean, who really cares? Well, why you should really care is 1 degree Fahrenheit increase in temperature equates to around 1 million square miles of permafrost lost. So you can see why and how that would feed back into that positive feedback in a really, really pronounced way. And that is an excellent comment from Arcus, possibly from Janet or Judy, is that positive feedback sounds like happy, positive feedback. And so it's probably something that needs to be rebranded in science, because it's definitely, in this case, not a positive thing that's happening. So for the rest of this presentation, now that we've done some background, we're going to focus on the specific project that I worked on when I was up here the most, which is a photo bio project, and you'll see why. This project is a really nice collaboration between 3 principal investigators who are all up here and that I've gotten to meet. The first is Dr. Byron Crump out of Oregon State University. The second is Dr. Rose Cory, who's sitting right next to me, out of the University of Michigan. The third is Dr. George Kling, also out of the University of Michigan. And each of these principal investigators has their own specialties that they bring to the table, and it fits together in a nice way to allow this project to be so broad. I should also point out that this specific photo bio project we're working on this summer is the work of Natasha Chrisman out of Oregon State University, and she's going to be using it as part of her PhD project. So she's a graduate student here who's been a really great mentor for me.
Okay, so yeah, so our specific goal, so that, you know, overall what we're looking at here is of this carbon coming out of thawing permafrost soils, because the Arctic is warm and the permafrost is thawing. What we're trying to understand is how much of this carbon is going to be turned into carbon dioxide, and what the time scale of that process may be. Is it 10 years, 50 years, 100 years, or is it going to be flushed out to the Arctic Ocean as dissolved carbon where it's not contributing to warming? And so what we're working on is based on one of our discoveries of a few years ago, which is really the importance of sunlight. So the conventional wisdom was that it's all microbes that are respiring, just like we take in carbon in food, we eat it, and we breathe out carbon dioxide, and that's called respiration. And the conventional understanding was that the main way that this dissolved organic carbon was getting turned into CO2 was just microbial or bacterial respiration. But we knew that this material, this dissolved organic carbon, is colored, meaning it absorbs sunlight. So we had the idea that there's an abiotic or photochemical reaction that's also important, and that's exactly what we found. So we found that sunlight on its own can completely oxidize dissolved organic carbon to CO2, and that's just as important as microbial respiration. And in addition, we found that sunlight can also bleach or partially oxidize this colored dissolved organic carbon, and that carbon is washed out to the Arctic Ocean, and so that's the blue part of this pie diagram here. And that's important because, again, carbon in that form may stay as dissolved organic carbon and not contribute to greenhouse gases.
So, an overview of what this PhotoBio project entails. The main goal here is to understand how specifically dissolved organic carbon is being broken down to carbon dioxide in Arctic watersheds. Why? The reason why is that current climate models cannot effectively account for permafrost positive feedback, simply because not enough information is known about how this process works. So the goal of this project and most of Dr. Cory's research is really just to figure out mechanistically, rate-wise, how fast this process is occurring, why it's occurring mechanistically, so that in the future, climate models can be updated to better account for positive feedback in the Arctic. And there are three critical questions that are being asked in this project. The first is, how is the chemistry of dissolved organic carbon specifically affected by, or how does that chemistry affect the metabolism of microbes, the microbes that are in the soils, the microbes that are in the water? The second question, which is the one that we're really focused on, is what does soil depth have to do with this? So does this process occur differently in surface mat soils versus permafrost soils? And there's a second part to that second question, which is really, what does sunlight have to do with this? As Dr. Cory just pointed out, her research from 2014 illustrates that sunlight is a huge part of the puzzle in terms of breaking down dissolved organic carbon to CO2 in Arctic watersheds. Well, interesting question. Are bacteria better able to metabolize organic material that has been exposed to sunlight? Are these two things working synergistically?
So yeah, Arcus, so someone asked the question, what exactly is microbial respiration? Again, it's just like us taking in organic carbon in food, and we use that some for energy, and we breathe some out as CO2. And what we're trying to understand here is how do microbes do that, and how does basically sun-brewed DOC respire differently? So it's kind of like thinking about, eating Snickers versus all brand, so Snickers bar might be, quick energy, and we maybe metabolize that quite differently than a bowl of all brand. And that's really what we're trying to understand up here. And so we have different kinds of carbon, how much of that will be turned into CO2, and how does exposure to sunlight change that process?
Yeah. And then question 3 is, how does community adaptation work here? So how does evolution play a part into this cycle? So are microbes at different soil depths differentially evolve to metabolize different forms of dissolved organic carbon? And how is climate change factoring into this? So I'm going to go through a little bit of the process that I've been a part of, which is, I feel really lucky that I've been a part of this entire process. When we got up here, we started the day after. This is our study site. It's in Naviat Creek, which is really close, just north of Toolik Field Station. And we're specifically working, that photo on the right represents the study area. It's on the wet sedge tundra. Tundra being the primary environment up here, no trees because of the short growing cycle and the cold temperatures. So that's the research team working out, walking out, possibly also working out at the study site. So the first 3 days that I was here, we spent digging soil pits. And this is a procedural flow chart that will kind of take you through. It's a little bit complicated, so I'm going to have Dr. Cory give you a high-level explanation.
Yeah, and I don't want to spend too much time on this except for to say that that the work that we do up here is difficult and or complicated, I should say, and soils are very complex. So if we go out and take some soil, how do we know that we've chosen representative soil? And so part of what you see here, just a snapshot, is that we have replicates. So we take multiple soil pits to try to capture the variability on the landscape. And then because we're looking at the effect of sunlight on how well microbes can respire this DOC to CO2, we have light exposed and then we have a dark control treatment. So we're always comparing in a treatment, we always have our appropriate controls. And that's really what's shown here in this diagram is trying to capture variability on the landscape and then also having the appropriate control for our light treatment.
So critically, two independent variables, light versus dark, and surface mat or active layer soils, that stuff that freezes and thaws with the seasons, and permafrost, the stuff that's permanently frozen. So the first 3 days that I was here, which was pretty remarkable, I spent helping dig huge pits in the permafrost. Because, as you can see on this side, there's 3 replicates, we dug 3 permafrost pits. And basically, we got a bunch of different stuff from the permafrost pits, but it was pretty tough work. You can see here on the left, once you get through that active layer, permafrost gets kind of like concrete because it's from frozen soil. So it's very hard. You can't just dig into it with a shovel. So we had to use a jackhammer to get down. We went down about 1.5 meters in each pit. That took about 6 hours for each pit. So that was spent each day digging down. And it was an interesting process because you could kind of discover different things in the permafrost. So for instance, these pictures down here at the bottom, that twig just looks like kind of an ordinary twig until you realize that it's been sitting frozen in permafrost for 9,000 years. They've done radiometric dating of the soils in this area, and that's how old the permafrost is. You also encountered a lot of different ice features. There were ice lenses, horizontal layers of ice that we would have to chisel through, also inclusions, little specific zones of ice. But we eventually got down to around 1.5 meters. And this is a cross-section of what a completed pit looks like. So you can see, I took this photo and sort of added on the dimensions. But up at the top there, so the top layer, vegetative matted surface soil, that's all active layer material that freezes and thaws with the seasons. We then typically had to get through a mineral layer that consisted of clay. And then finally, around about 1 meter in depth, we got down to permafrost, which is pretty highly concentrated organic soil that has been frozen for at least 2 years or more. The second step, did you want to add something?
Just that there are a couple questions here.
Sure, yeah, go ahead.
So, yeah, someone from PolarTREC wanted to know how many different representative soils are in the Toolik area. And one, well, this is all permafrost here. But then what distinguishes some of the features of the soil is really the landscape age, so the time since last glaciation. So we have some areas here where we call the landscape and the soils on them relatively young, meaning they were glaciated like 10 to 60,000 years ago, and then we've got on the other end soils that or landscapes that it's been a long time since glaciation, so say greater than 100,000 years. So that results in different types and chemistry of the soil, so we make sure to sample those different soils. And then someone else, oh yeah, go ahead.
That's my cousin Kelly. Great question, Kelly.
Great.
You mentioned that organic material is colored and that it's bleaching. Is the colored material changing proportionally, or is the colored material what is being converted to CO2? Excellent question. So it is actually the latter. It's the colored material being converted to carbon dioxide. One of the common reactions that happens here that Dr. Cory has figured out is decarboxylation, which is basically part of that huge, one of those, for instance, that huge molecule of lignin that I showed you, a part of that breaking off and becoming carbon dioxide. So in a lot of instances, a single molecule can generate multiple molecules of carbon dioxide, depending on the oxidative process and stuff like that. But in its simplest form, oxidation in organic chemistry is an increase in the number of carbon-oxygen bonds. And so while one thing might be getting reduced, the oxidation process is generating carbon dioxide. did you have anything to add?
Okay.
Okay. Bruce asks, no mosquitoes. Mosquitoes are pretty rough now, but at the beginning of the season when we were digging these pits, the mosquitoes weren't bad. I was mainly concerned about the rains and stuff. So once we had these big pits excavated, sampling took place. And remember I mentioned that there's the independent variable of soil depth. So we took 2 categories of sample. One at the surface mat layer of soil, that's the active layer, and you can see on the left there, Dr. George Kling is chiseling out a sample for his graduate student, Carl, who's collecting it in a bucket. And then on the right, Carl is drilling into the other category of that independent variable, the permafrost layer. So we got a ton of soil is needed because remember this experiment is on dissolved organic carbon. So you have to have a ton of hard material from which to leach a bunch of dissolved organic carbon. And that's the 3rd step. And they say permafrost tea up here a lot, and this step was really brewing permafrost tea. On the left there, that's a bucket of soil, either surface layer soil, active layer, or permafrost soil. And we added a bunch of water. There are tons of replicates here. Added a bunch of water, let it sit for 72 hours. And on the right, that's what it looked like chocolate milk.
And remember, we're doing this to mimic what happens naturally in that as permafrost soils got deeper, snow and rain moves through the soil and leaches that solid soil material and flushes it into water in that color. And so that's what we're doing here. We're mimicking that process in the lab.
I bet it doesn't taste like chocolate milk. I have not tried it. I have smelled it, and I'll get to that. Dr. Cory has apparently consumed permafrost tea, which we'll have to talk about later. So part 4, as you can see, that kind of muck on the right is you got to filter that because you got to get out all the soil material. So part 4 is filtration, and this is graduate student Natasha Chrisman filtering, and this was a really cool process for me because look at the dramatic color change. You're passing this chocolate milk sludge looking stuff through a 0.2 micron filter, and that gets out all particulates. It also gets out all microbes. So look at what it looks like when it comes out. It's this light yellow looking material that really consists of water and the dissolved organic carbon that was in permafrost or surface material soil, permafrost tea. So this was a really cool looking material. It had an interesting smell to it. I posted a video at some point on YouTube. It's on the PolarTREC channel if you want to check it out, where everyone has a different interpretation of the smell of this stuff. To me, it smells like kind of manure in a cold, dark basement, but everybody's got a different thing that they think it smells like. But it's a really cool material. And then Dr. Cory's favorite part 5, the photo exposure part. So this is where we introduce the 2nd independent variable, that's sunlight. So we transfer we're going to call this the filtered leachate at this point, the permafrost tea. You transfer it to bags and tubes, and you set it outside. And you hope that you get good days. And if you don't get good days, you got to pull it all back in and wait for a good day. A good day meaning lots of sunlight. You can see there on the right, those are samples of permafrost tea being photo exposed to sunlight in that cooler in the back that we wrapped on the outside and on the inside with aluminum foil. Those are the dark controls. So if you remember that diagram that Dr. Cory was explaining where there was a light and a dark category. So we have light exposed and dark controls. So this went for 24 hours in full sunlight, leading to step 6. Remember that this permafrost tea has no microbial content. And so to study how microbes react to sun-brewed versus dark control permafrost tea, you have to reintroduce microbes. And to reintroduce microbes, we use that original chocolate milk stuff. We just passed it through a much coarser 0.5 micron, I think it was 5 micron filter, to leave the microbes in there, and then reintroduced it to either the sun-brewed or dark controlled DOC and let it sit for 5 days. And during this 5-day incubation, there are tons and tons of sample points. What they're doing is they're analyzing very critically how microbes are responding to the dissolved organic carbon, whether it be sun-brewed or not, et cetera, et cetera. And Dr. Cory's going to get into that on the next slide.
Yeah, and someone asked, how many good days have we had for this part of the project? Many. We've been really lucky. The reason we come up here in June, though, is that June tends to be the sunniest part of the season up
Solstice.
here. And it's also solstice, as Dave points out, means it's the longest day of the year. So we, if the weather's good, we have the most sunlight for our experiments. That's when the sun angle is highest and most intense. So for photochemistry, we are always up here in June. And sometimes you have bad weather, but we have had mostly good weather for these experiments, which is great. So yeah, what we're looking at, of course, is how is the DOC changing in chemistry when we expose it to light, and then we're looking at how the microbes are responding to that change in their food, basically the change in what they're eating and respiring. So of course, we're measuring the production of CO2, both by sunlight and by the combined actions of sunlight and microbes. And you see my research technician, Katie, measuring CO2 in the upper left corner there on our carbon dioxide analyzer. And then my new PhD student, Emma, is in the top right graph. She's measuring how the light absorbing properties of DOC are changing, like how that DOC is bleached. And that's a property you are all familiar with if you leave your colored clothing out in the sun. You know, during the summer in Texas, you probably noticed that your clothing or your car paint will bleach. And that's what is happening to the dissolved organic carbon. And we quantify that on a very sensitive instrument up here. And then we look again we look at how the microbes are responding. Basically, how much more or less of the DOC do they respire or turn into CO2? So again, we use the CO2 analyzer for that. And then we look at also the microbial community in terms of who's there and what are they doing. So we look at their genetic data. And we also do metatranscriptomics to look at what genes are they expressing in the light exposed versus dark control. And that tells us a lot about how the sun brewing of DOC affects how they can metabolize or respire different molecules and turn some into CO2 and why they don't turn others into CO2.
I've been amazed at the amount of really, really high level technology that's up here in the Arctic. And this is a gas chromatograph right behind us, which is incredible to have up here. And all of these machines are really high class stuff. And that was one of the things that shocked me the most when I got up here is, wow, they do a lot of their analysis and things like that on site. Really amazing. So this study is, oh, how do you deal with no darkness? We can answer that. Let's answer that once we finish, because that's a good question. This study is ongoing, so there's no real results from it yet that we can present. But I wanted to take you to some results that are very recent from one of Dr. Cory's most recent graduate students that are pretty amazing.
Yeah, so this is my work for my PhD student, Colin Ward, on which our current work is based. So we're testing some ideas that resulted from this work. And the bottom line is that when DOC is exposed to sunlight or sun-brewed, just like sun-brewing tea, microbes prefer to respire this DOC. So you can see on the graph here, if you look at the dark controlled DOC in the darker red, and what you're looking at is the CO2 produced from microbial respiration, or they're eating this DOC and turning it into CO2, and how much they turn into CO2 is plotted on the y-axis for different replicate DOC samples. And what you can see is if you compare the dark versus the light exposed DOC, that a lot more of the light exposed DOC is turned into CO2. So about 2 times more. Microbes prefer by a factor of 2 to turn this light exposed or sun-burned DOC into CO2. So the implication here is that as permafrost soils thaw and this carbon is increasingly exported to sunlit surface waters, again where it's seeing sunlight, that more of this carbon is going to be turned into CO2 that's released to the atmosphere. Once it's in the atmosphere here, it is mixed around the earth and contributes to warming everywhere. Right, so that's why so to understand how much this carbon dioxide can contribute to warming everywhere on Earth, or this Arctic amplification, we need to understand exactly what molecules the bacteria are respiring, because then we could look at a chunk of soil and say this much carbon will be turned into CO2 over 10-year period, 50-year, 100-year, or 1,000-year. And that's the kind of information that we need to have if we're ever going to think about constraining CO2 emissions. We have to understand the component that's going to come out of the Arctic no matter what we do, what decisions we make. So it really does come down to the details, which is what's shown on this graph, which is pretty complicated stuff here. But basically what we find, I'm not going to explain the graph so much, except to say that the compounds that are broken down by sunlight, or sunlight turns these DOC molecules into compounds that the bacteria really like to consume. It's the compounds that they would consume anyways. And so sunlight is basically making more for them. You can imagine, Dave has a good way of explaining this. It's kind of like going to your favorite buffet restaurant, and you pick out certain things at the buffet, and then someone comes and puts more into your favorite food selections at the buffet. That's what sunlight's doing. It's making more of the food that the microbes like to consume already. And that's why we think they respire more of this DOC to CO2.
So why is this such an amazing result? Well, Dr. Cory just pointed out that this research leads to the suggestion that microbes prefer sun-brewed organic matter, which means that microbes and sunlight are working together synergistically in the Arctic. So that's huge, because that allows for a complete reevaluation of this permafrost positive feedback, because sunlight should no longer be considered a small part of the puzzle, and it should no longer be considered a part of the puzzle that's working separately from microbes. They're actually working together. Sunlight is manufacturing organic material that is easier for microbes to consume. And that's important when we take that to predicting future climate change and updating our climate models. So that's the real output of this, is that we hope to get a better climate model so that governments can more effectively regulate emission standards and stuff like that. So that's really the nuts and bolts of what's going on up here. There are a lot of questions that we have sort of flowing in the group chat, and I will ask Janet for protocol on how we should answer questions. Would you like for us to just go up to the top and start going in order, or?
Sure. And I think why don't you start asking questions, and if you need to reference back to some of your presentation, you can do that. If it turns out where you aren't referencing it as much, then stop sharing so we can see your faces better.
So Regina, I'll address this one because it's very related to just what we talked about right here. And Regina, this is a great question. So do you think that the heat of the sun influences the results? So if we kept the DOC in the dark but heated, would we expect similar results? And it's a great question, and the answer is no. You need the high energy. So the sun, of course the sun puts out a range of wavelengths of light, from the UV, which is very high energy, short wavelength, to the visible, to the IR, which is really heat. So long wavelength and low energy. And while we feel that heat from sunlight, it's not enough energy to break down organic molecules. And you could try this at home, maybe with your students in your classes. Try, carefully heating up some colored clothing, and you won't see any change. Whereas if you leave that colored clothing, like a shirt or a hat, out in sunlight, it's going to bleach, it's going to break down. And that's because you have that high energy wavelengths from light, the UV, and to some extent the visible light. But the IR is just not enough energy. You don't get any of that thermal breakdown or thermal degradation.
Great question. Dave, the science is complicated but important. How will you share what you are learning in your classroom with your students? Well, that's the real goal of why I'm up here, is to communicate this science with the public, also with my students. And I've had quite a few ideas. I think up here I've been kind of just sort of a sponge, absorbing the different methods and things that have been going on and thinking about it. And really, when I get back, I'm going to be able to cement my ideas more formally. But one of the coolest things I've seen involves this guy, which is a gas chromatograph. And we actually had donated to us at Lhasa High School a gas chromatograph from the University of Texas. So that's allowed us to do a lot cooler experimentation. And I had been doing a lab that I had wanted to change quite a bit that involved students identifying unknown alcohol mixture or solution via gas chromatography. Since I'd been seeing them go out in the field and collect dissolved gases from water samples up here, I thought that a very interesting thing that I could do, my students go out to different parks and preserves in the Austin area as part of their biodiversity projects already. I could have my Planet Earth students go out and collect different water samples from different parks and preserves in Austin and come deliver those to my organic chemistry students, who would then go through experimental process of grabbing out from those samples the dissolved dissolved carbon dioxide and measuring it on a gas chromatograph to get both how much carbon dioxide is in it and what other things are in it, like methane and stuff like that. And that can be related, even though we're doing it in Austin, it can be directly related to research that's in the Arctic, because soils are different everywhere. And as Dr. Cory mentioned, dissolved organic carbon is present in waters all over the place. So that was an interesting idea that I'm still formalizing, but that I think I'm going to go with for my organic chemistry and Planet Earth students to go from there. What findings have most surprised me? I was definitely not aware of how important sunlight was in this equation. Just like Dr. Cory, and I've talked to her about her research in 2014. In fact, I asked her at one point what sort of her best experience in the Arctic was, and it was actually the realization of the importance of sunlight in this puzzle. It's incredibly important. It could be argued that sunlight is even more important than microbial processes and breaking down dissolved organic carbon in the Arctic. So I, one, did not know it was that sunny up here during the summer, and two, I did not know, I was shocked to see the importance of sunlight from reading Dr. Cory's papers. Other questions? This is a good one that I actually don't know the answer to. Have these findings been incorporated into the major climate models yet? From Bruce. Hi, Bruce.
Yeah, no. And so there's the climate models are just starting to include feedbacks from permafrost. So they're just starting to include Arctic amplification, but none at the level of the kind of detail that we're trying to include. So it's a very crude sense of this much carbon may thaw, this much may get oxidized to carbon dioxide, but you know, they're guesses, and while they're good educated guesses, they're to really understand how this Arctic amplification is going to feed back climate change on Earth, we have to know and understand what are the controls on the conversion of this organic carbon into carbon dioxide, which is what we're working on, so that we can put these processes into models and have better predictions. So that's the goal, but we're not there yet.
There's a question: as sea ice melts and more water is open,
yeah,
would you expect to convert even more DOC to CO2?
Yeah, that's a great question. That's exactly correct. So this, and not just in sea ice, but here on the Arctic landscape where we're working, we work in what's called a terrestrial system, but there's a lot of water on the landscape. This is basically a giant wetland. So there's a lot of lakes and small ponds, like more than 50% of the landscape is covered with water, and that's fresh water up here. And we are seeing earlier ice out on lakes and streams across the entire Arctic, not just the Alaskan Arctic, but Siberia. And that earlier ice out in what's spring, it's called spring up here, that's May and June, that happens at the time of year when the sun angle is highest, and so the intensity of solar radiation reaching the surface here is the highest. And that's also the time of year when snowmelt flushes in all this colored dissolved organic carbon into waters, and so you have this perfect storm of factors that are creating or could increase the amount of CO2 coming out of the water. So for example, this summer, we had the earliest ice out on Toolik Lake here, the lake at our field station, on record by a long shot. So ice out happened on June 4th, basically the day after we got up here, which is very early. And so, you know, basically we're just watching the CO2 come off the lake effectively from photochemical reactions. But the same thing is true for sea ice. So as rivers bring all this dissolved organic carbon into the coastal water, and sea ice is getting thinner, and there's less and less sea ice each winter, there's huge potential for this organic carbon fresh from land to coastal waters to be turned into CO2.
And as related, Hey, it's my mom. My mom, Lisa, asks, any way to drive more of the DOC into the ocean? And because I noted in quite a few of your papers, you were talking about the shallowness of Arctic headwater streams and how that's a factor. So that's a good question. I'll let the expert answer.
That's a good question. I don't know that there's anything we could do to control that, because effectively, if we can bury this DOC in the ocean, it would be sequestered and not carbon in sequestered form, not contributing to greenhouse gases. There are potentially changes in hydrology or changes in river flow, the timing of water moving across the land and into the ocean that may influence how much of that DOC could get sequestered in the ocean. And there are lots of people working on that component of the research up here. Whether or not we can control it, I'm not sure. I don't think we can, but we can try to understand it. So at least we know what we're in for.
You've done research on headwater streams as compared to like tertiary primary streams.
Yes.
And is there more DOC in headwater streams? And it's just all getting by the time it reaches the ocean?
Right. So that's right. So in smaller headwater streams, there's generally higher DOC concentrations, and then the larger rivers and streams, and in part that's because of the photochemical and microbial oxidation of this dissolved organic carbon. So turning it into CO2 as it's moving through these small streams into larger rivers on its way to the ocean. Is that what you're asking?
Yeah.
Okay.
Yeah.
Good.
See Cory. See Cory, is that your?
Claire, that's my sister.
Oh, that's Cory's sister. Hello. I'm a big fan of the PolarTREC program, as I've heard about it from Rose for a long time. David, appreciate your dedication to teaching and students. Thank you. It is important to get this information out to students, but how do you share with fellow teachers? Good question. I've done a lot at the school and district level. That's pretty easy because I know all those teachers. So I can do, talks at my school for our professional development meetings throughout the year on PolarTREC and on this research. And I also know a lot of people at the district level. My way of reaching teachers across the nation is I've submitted an abstract to the NSTA, which National Science Teachers Association National Conference. And I went a couple years ago to that conference, and just all sorts of teachers attend, and they're usually people that are pretty dedicated to the practice. It's the best way that I've found to network with other teachers. So I'm going to be presenting at that, and then hopefully with Dr. Cory at a lot of other teaching focused meetings. There's one that our school specifically participates in for STEM schools. And those meetings really create a nice context for talking about this type of stuff. So that's my plan for now, but in general, teachers are very receptive to ideas, especially if they come with lesson plans. So I want to go to those conferences and things like that with some lesson plans that I can actually share with teachers. Bruce and Regina have provided a great model for this. They've been PolarTREC teachers with Dr. Cory in the past, so I can look at the work that they've done. But it really helps to say, hey, this is really cool research. Here's how you can implement it in your classroom. Here's a packet you can take with you that has this lesson plan. These are where my resources are online before you get into that game. Effectively, with the recent warm temperatures in the north, is there technically a loss of permafrost? If so, how would you measure that, something that is no longer permafrost? Good question.
Yeah, that's a good question. So, across the Arctic, there are measures of change, different kinds of measures of change in permafrost. So, one is thaw depth. So just jamming a steel rod into the ground until it stops, and it stops when the ground is frozen, and so you can then measure how deep you push the rod in, and that measure of depth is a measure of depth to permafrost thaw. And so of course that changes over the summer season as the soil thaw out in the active layer. And so people are measuring thaw depth across the Arctic, and in some locations that is increasing. So you're thawing deeper and deeper and deeper into the soil over the summer. Another way that people are measuring changes in the permafrost is measuring temperatures of the permafrost. So there's records going back about 40 years or more at very deep, like I think it's to 20 meters below the surface, that show increasing temperature in the soil. So not yet above freezing. So technically that's not a loss of permafrost, because permafrost soil means that it's been frozen consecutively for 2 or more years, and so the temperatures are still below zero or below freezing, but they're warming up substantially over the past 40 years across the Alaskan Arctic and other places in the Arctic as well. But there's actually an even more sensitive measure of change in permafrost, and that's the geochemistry of what's being flushed out into the fresh waters on this landscape. So we know that the chemical signature in permafrost or frozen soil is different than what flows out of the annually thawed active layer. And then we can actually see measures of that change in the water around here. And there's no other way to get that change other than by thawing deeper into the permafrost soil layer. And it's not just in the Alaskan Arctic. People are measuring this, finding the same results in the Canadian Arctic and the Russian Arctic as well.
Let's see. Was someone trying to say, were you trying to say something?
It looks like you have one more question there from John.
Okay. Would there be, from John, I'm not sure if you mean John, my father, or John, my uncle, but whichever John, hello. Would there be a difference in using a 0.2 micron filter versus a 0.02 micron filter? Yes, there would be. I think it would be very difficult to get something through a 0.02 micron filter. Do they even make 0.02 micron filters? I don't even know if that's able to be engineered.
Yeah, no, but it's really interesting to think about. If you think about what does it mean for something to be dissolved, we use that term. There's different meanings for it scientifically versus everyday life. If you're say dissolving some electrolytes or something in water, like a salt tablet, and there's really no chemical definition of dissolved. It's an operational definition of what passes through a filter. So scientifically, what we mean by dissolved is just anything that passes through a 0.7 or a 0.45 or a 0.2 micron filter. And beyond that, you just really can't get stuff through. It becomes really difficult. So we kind of go with this operational definition of what works, what we can do. And in addition, we know that most microbes are bigger than that. So if we want to filter something to remove microbes, that does the job.
0.2 was very difficult to get stuff through after a bit. So you saw that chocolate milk stuff that we got as permafrost leachate. We passed that through a coarser filter that removed all the sort of larger scale particulates, went through that pretty easily. And then we pumped it through these 0.2 micron filters. And after a while, the flow rate was significantly slower. So yeah, I don't think they make filters that are much smaller than 0.2 microns. But good question.
So I'm going to start wrapping up things, and if you have more questions, feel free to post them to David in the journals, or send them through an email to him. But I just launched a poll for those of you that are participating in this webinar tonight, so please take a moment and vote, or give us your feedback about this PolarConnect event. And in the meantime, while people are doing that, I really want to thank you, Dave and Rose, for your presentation. It was great. Very complicated science. Every time I hear it, I learn just that much more. And I'm just always in awe with the research that you do, Rose. It's like really amazing. And I'm really, Dave, you did a fantastic job of translating it for the rest of the world so we can understand it and making it very useful for us to grab onto and understand better. So thank you very much.
Thanks to you all, and yeah, thanks to Janet and Judy, who've been amazing throughout this PolarTREC process, and of course to Dr. Cory, Dr. Kling, Dr. Crump, all the grad students and technicians I've worked with here. This has been a really awesome and educational experience for me. Thanks to you all for joining and learning about climate science. It's very neat.
So how much longer are you going to be there? What's your plan? And
We both leave this Saturday.
Yep.
We leave this Saturday. So it's been sweet. I've gotten to know this place, and it's a really fun spot. I've made some friends up here, but I will be meeting my wife Ellen in Fairbanks, and we're going to do a little touring around of Alaska. So that'll be an awesome experience in and of itself. So, yep, this Saturday. Got a couple more days here.
that's amazing. Hard to believe that the time is coming to an end.
I know.
I'm excited for you, Ellen. You're gonna love it.
Yeah, I know. So, yeah, so we thank you everybody for sitting in on today, and we look forward to the last little bit of your your time there, I should say, up at Toolik, Dave, and have safe travels back.
We'll do. We'll do. I'm going to post a lot of cool bird and wildflower photos on my research journal these past couple days. So got a lot of good videos. So stay tuned.
Yeah, and have a good visit with Sarah. I know you're going to see her. So.
Yes.
Yeah.
Cool.
All right. Well, thanks everybody. If you're joining, I'm going to stop the recording. If you're family or whatnot, you're welcome to turn on your videos and say hi and all that good stuff to everybody.